Hello, everyone. I want to thank you all for joining us today for ALS Webinar Wednesday series. My name is Kelly Hall, and I'm a marketing coordinator for ALS North America. Our presenter today is Ranil Fernando. Ranil received his Bachelor's of Science degree in microbiology and a minor in chemistry from California State University in Long Beach. Ranil is a member of the American Chemical Society and American Society of Microbiology. He has over 35 years of industry experience in managing quality control testing laboratories. Now he serves as the general manager at our ALS-BioScreen laboratory in Torrance, California. Today, Ranil will be discussing stability studies in pharmaceutical and personal care products. Ranil, thank you very much for taking your time to deliver this presentation. I will go ahead and turn it over to you.
Thank you, Kelly, for the introduction. Hello, everyone. Thank you for joining the ALS Webinar Wednesday series. And the topic for today is stability studies for personal care and pharmaceutical products. It is my pleasure to provide this information to you. So let's get started. First, I do have a disclaimer. This information is my professional opinion, and it's not necessarily that of ALS or BioScreen testing. And these are the scope of the presentation, and we'll cover the different sources of information, the objective of doing stability studies, glossary of terms, different types of studies for pharmaceutical products, some reasons for stability failures, data reporting, and personal care studies as well, and then a detailed list of the references. So first of all, rather than just do the references at the end, I thought it was very important to discuss where the information for the webinar came from. And it has been gathered from these sources: ICH, WHO, FDA, EMEA, ISO, PCPC, COLIPA, and various pharmacopoeia. And I'll go into a little bit about each organization. Majority of the information is from ICH, the International Council for Harmonization, and they focus on 4 areas of standards: quality, safety, efficacy, and multidisciplinary information. And a link to their or a screen capture of their website is listed below. Pretty easy to remember, ich.org. And this is another screen capture of the quality attributes. And there are 14 different standards. And first one is Q1A, Q1F for stability testing of new drug substances and products. And what's important to note is that right now it's at status 5. It went through status 4 in February of 2003. And you can see the countries or regions that have adopted this guideline. So Brazil, Europe, US, Singapore, Canada, Republic of Korea, Japan, China, Switzerland, and Taipei. So for each document, it'll list which countries have adopted it. So you can see most of the major regions or enough countries have adopted this document. The next sources of information is the WHO. I'm sure you've heard of WHO, and their website is WHO.int. But in their medicine section, you can see different regulations and publications that will allow you information. Some of those are listed at the end. Followed by the FDA, the U.S. Food and Drug Administration. Their website is fda.gov, and a quick screen capture of the different topics. And again, it's searchable. All of this is freely available for anyone to download. What you'll find in terms of stability for FDA is that they obviously refer mostly to ICH since it's been harmonized and adopted. Next is the European Medicines Agency. Again, the website's listed there. And again, most of the references link back to ICH. There's some information from the ISO as well, especially for the personal care side of things, and there are ISO.org. Next, I listed 2 trade organizations. the Personal Care Product Council for US, the PCPC, and the European counterpart, the European Cosmetics, Toiletry, and Perfumery Association, and their websites are listed here as well. Now, because they are trade organizations, the information that they provide has to be through a membership to the organization, so you do have to pay for that. Next is the various pharmacopoeia. I've just listed a couple of them here, the USP and the European Pharmacopoeia and their websites. Again, you do have to be a member or join the organization in order to get the information. So moving on to our topic, what is the objective of performing stability studies? The definition, in a sense, is listed in the ICH document, but really it is to assist in product development, determine properties of the product, establish the storage condition, determine a retest or a shelf life of the product, a shelf life period, and determine product quality. And obviously to meet the regulatory requirements by the various regions or countries. We will go over some of the terms, and starting in alphabetical order. One of the first ones is accelerated testing. By the way, these are not all the terms that are listed in the ICH documents. I've just only picked a few. Obviously, one of the goals with stability testing is to perform it in a quick manner, and you do that by increasing the temperature of the storage time, and by hopefully changing the physical or chemical degradation of the product. Normally, we'll cover that in a few slides afterwards, but it's normally either 5, 10, or 15, or even 20 degrees above normal storage condition. Next topic is bracketing. And this allows clients to basically perform studies at the extremes of the dosage forms rather than perform testing on every single different package size or package strength, and that is a cost savings. Climatic zones, there are 4 zones that are recognized by ICH, and we'll cover that in a further slide. Commitment batches or registration batches, this is something that the agencies, whether it's FDA or European agency, will require a company to commit to for a new drug or a new drug product. And normally, because by the time you're registering, you may not have all of the data available. You might only have 6 months to 12 months of data. And by committing to perform usually minimum of 3 or maybe more batches, you're letting the agency know that you're going to go through and perform all the needed testing. The container closure system, this is the packaging components that together contain the product and protect the dosage form. The dosage form, obviously, I'm sure you're all familiar with the different types of pharmaceutical products, the tablets, capsules, solutions, creams, and so forth. The definition for a drug product is the dosage form in the final immediate packaging intended for marketing. So normally the registration batches are done in the final dosage form for the final product. The drug substance, the API, this is the unformulated drug substance that may be subsequently formulated with excipients to produce a dosage form. And for excipients, it's anything other than the drug substance in the final dosage form. Expiration date. So we're all familiar with the expiration date. It's not just for pharmaceutical products, food products have it. So it's usually the date that the product is good until. And normally, you should not use the product after that date. Long-term testing. This is normally the storage condition of the product. So if a product is meant to be sold at room temperature, then your long-term testing would normally be done at that temperature. The shelf life of a product, also referred to as the expiration dating period. So this is the amount of time that the product is good until. It could be 2 years, 3 years, maybe even longer, and that it meets the same quality attributes as when it was originally manufactured. Stability in the USP, this is defined as the extent to which a product retains within specified limits and throughout its period of storage or use, shelf life, the same properties and characteristics that it possessed at the time of manufacture. Next two slides cover the term specification. They're slightly different between ICH, Q6, and Q1. Here, it's listed as the specification is defined as a list of tests, references to analytical procedures, appropriate acceptance criteria, which are numeral, limits, ranges, or other criteria for test is described. It establishes the set of criteria to which a new drug substance or drug product should conform to be considered acceptable for its intended use. And it's possible that there will be some tests that are performed that are not necessarily performed at each interval. An example would be container closure integrity, where you might only do that at maybe beginning end or beginning middle end, or even some microbiological test. Slight differences in terms of specification in the Q1A, really, but it really covers the same thing. For release product, it's the combination of physical, chemical, biological, or microbiological tests and acceptance criteria that determine the suitability of a drug product at the time of its release. But if it's for a shelf life, it's really the same wording, except that it's good throughout its retest period, and that a drug product should meet throughout its shelf life. So let's move on to the different types of stability. First would be, let's say, chemical. Is the drug product or drug substance retain its chemical integrity, label strength within the specified limits? If you're having a product such as acetaminophen, is it going to be at the same strength throughout its shelf life, and is it going to have any degradants that are increasing over time? It could be obviously the physical stability of the product. Does it retain its appearance, the color, the odor, the melting point, uniformity of dissolution, any of the physical attributes? Does it look the same, behave the same throughout its shelf life? Obviously, microbiological, this could be the antimicrobial effectiveness for products that are preserved, or for products that are sterile. Does it remain sterile throughout its shelf life? And the goal of doing the chemical and physical and microbiological stability is to prove that the therapeutic, effect remains unchanged from day one to for year 2 or year 3, as well as the toxicological. There's no significant increase in toxic byproducts. And when is stability studies conducted? Obviously, at pretty much every stage of the product life cycle, at a very early stage during R&D, and especially at that point, you will be performing stress testing as well as accelerated testing. During pre-formulation batches, you'll be performing stability studies, and any scale-up batches as well, and especially to do stress testing and accelerating at that point. At stage 4, the registration batches, at this point, you would be performing not just accelerated, but especially long-term testing as well. And the minimum is 3 batches, but many companies perform many more batches than just 3. And obviously, once a product is released, it's normal to perform at a minimum 1 batch per year for each dosage and package size. And finally, there may be follow-up stability studies depending on the changes to the product. It could be changes to the excipient, it could be changes to the manufacturing practices, or changes to the manufacturing location. Just to go over, obviously, some of the dosage forms that will be tested: solid products, liquids, semi-solids, lyophilized products, as well as combination products. This would be an example like EpiPen, which you have a drug in a device, basically. Going over the various climatic zones, the ICH has defined 4 climatic zones. The 4th one is actually has a part A and a part B, but these are your temperate climate, the subtropical Mediterranean climate, hot and dry, and the 4th is hot, and humid, and hot and very humid. And the last column under the storage condition, you would normally store your products at this temperature and humidity condition. I wanted to reference back to the WHO Annex 2 Technical Report 953. So this provides the storage condition for each country. It's broken up by the various regions. So you can see there are different temperature and humidity conditions there. And I've listed the countries that were in the signatories to the ICH. And you can see in general, most countries go with 25 C, 60% relative humidity, or 30 degrees C, 65% for things that are at the zone 2 storage condition. Let's talk a little bit about the not just the storage condition, but the duration as well. So this would be a product that's typically stored at room temperature. Your long-term would be at 25 C at 60% or 30 C at 65%, minimum 12 months. Many times this would go to 24, 36, or even 48 months. The intermediate would be 30, 65. And the accelerated would be at 40 degrees at 75% relative humidity. If the product was meant to be stored in a refrigerator, obviously the long term would then be at the refrigerated condition, 5 degrees plus or minus 3, and the accelerated would be at 25 at 60%. Many times these kind of products do not fare well at the accelerated. They're just not going to be stable enough, and it might only be good for a month or even less. Next would be if it's stored in a freezer. Obviously, you would store in a freezing condition at minus 20 plus or minus 5. If you have a product that's a semi-permeable container, which might allow a loss or gain of moisture, so you do want to be careful that these are then stored at the appropriate storage condition. The relative humidity would be different. So you can see 25 at 40% or 30 at 35%. The intermediate is high, 30 at 65%, which is the same as before. But the accelerated, again, the humidity would be much lower, 40 degrees, not more than 25% relative humidity. Let's go over the requirements for the frequency. The guidance says that for long term, it's recommended that every 3 months for the first year, every 6 months for the second year, and annually thereafter. So normal intervals would be initial 3, 6, 9, 12 months, 18, 24 months, and obviously 36, 48, and other months as needed. For the intermediate condition, at least 4 time points. So here you would do 1, 2, 3, and 6. So 1, 2, 3, 6, maybe add 9 or 12, depending on what length you're trying to achieve. And if it's accelerated, the guidance is to have at least 3 time points. So normally you would do 1, 3, 6, or possibly 1, 2, 3, and 6 months. Selection of tests. So it really depends on each product type, and I'll go over into a little bit more detail in later slides. But if it's a brand new chemical or drug entity, and during the R&D phase, it's important to come up with the tests that are the best indicators of the quality of the product. And these, again, could be chemical, physical, biological, or microbiological tests. And then if it's a monograph product, it's in the USP, EP, or the monograph, it's normal to perform those tests as well as any others that are predictive of the products, any changes that might occur to the product. So let's talk about an API. And so these are some of the common tests that would be performed to determine stability of a drug substance: physical, appearance, color, particle size, maybe identity test, chemical tests for the assay and degradants. At this point, you should have all the stress testing performed, such as bioxidation, photolysis, acid base, and you would make sure that the assay is able to pick up any degradants, the moisture content, any residual solvents, or impurities, elemental impurities, or any other monograph test for that particular drug substance, as well as any microbiological quality, aerobic plaque count, yeast and mold, as well as any other specified pathogens. If it's an injectable product, again, you would have some physical tests, possibly most likely container closure integrity, pH. Again, assay and identity would always be performed, and possibly some antimicrobial agents if they're in the product. And then for sterility, endotoxin, and particulate matter as well. For a lyophilized product, somewhat the same test as before, except you'd want to be looking at the reconstitution time of the lyophilized powder, appearance of the powder. Is it a fine powder? Does it have maybe, have different types of particles, or does it look the same? Again, container closure of the packaging, and the rest of the tests are somewhat the same. If it's an oral solution, you would perform similar tests as well as taste. You want to make sure that the taste doesn't change over time. And then for chemical assay identity, degradants, if there's any antimicrobial agents, as well as the microbiological testing. If it's a suspension, you would do similar set of tests, except you might add some redispersibility of the product, any of the sizes of the particles in the suspension, or rheological properties of the product, in addition to the normal chemical and microbiological tests. For inhalation and nasal products, you might be looking at the spray pattern, how much is dosed at each spray, a possible microscopic evaluation of that, and again, the similar chemical and microbiological tests. For tablets, obviously, you would then be adding tests such as disintegration, hardness, friability, dissolution time, as well as moisture content. You would have less tests here, primarily assay identity. And again, the residual solvents, elemental impurities, those might only be done, again, just maybe at the beginning of the study. And obviously, the microbiological tests as well. Gelatin capsules, you would add dissolution time probably and moisture content. There wouldn't be need for disintegration for these type of products. And for soft gelatin capsules, similar set of tests. You probably want to make sure that the capsules are not leaking after storage. If it's an emulsion, you would be adding viscosity, pH, specific gravity, solids or moisture content, and similar tests to the chemical and microbiological areas. I will touch on this very briefly. This could be another topic for a webinar. Obviously, it is expected that all of the methods that are used for testing of pharmaceutical products or over-the-counter products be validated. ICH-Q2 as well as ICH-Q3, A through D cover this topic. It is expected that the method is able to pick up any degradation or impurities in the product. And so these would be done at the R&D phase or pre-formulation phases. Same applies for any of the microbiological methods as well. And it is expected that method suitability be performed to rule out any false negative and in some cases false positives. Another topic of importance is the photostability of either the drug substance or the drug product, and it's normal to perform these at early stages, normally of the drug substance, and then if it's the drug product, you would be testing this in the immediate container. You might test it with the container wrapped in foil to have as a control, and then obviously in the final marketing package as well. This is the reference to the photostability. It requires that there be 1.2 million lux hours of exposure, as well as 200 watts of hours per square meter exposure at the visible light and the UV light. And normally this is done in a photostability chamber. I'm not going to go too much into the requirement. There's a method for measuring the exposure by using a quinine chemical assay, and they give you a couple of options, option 1 and option 2, on how to do that. In terms of the photostability of a drug product or drug substance, there's a flow chart that dictates how the product should go through the process. Obviously, you're exposing it directly, which is the API, and you're looking to see if there's a formulation change, and if there is, then you would have to figure out, does this product need to be packaged in a packaging that's impermeable to light? If there's no changes to the product, then you would just package it in normal container, and you would do this stepwise in each of the different containers, the immediate pack, the marketing pack, and determine if there's any changes needed on the packaging. The next thing to consider when designing a product and performing stability is the container closure system. So it's obviously very important to perform the testing in the final dosage form in the package and container closure system that's going to be marketed. There are a number of ways of performing the container closure testing. The USP Guidance 1207 Package Integrity Evaluation for Sterile Products is a good document. There are different mechanisms such as dye immersion, microbial leak test, or there's a helium leak test by LCMS that'll tell the company if the product is packaged appropriately and if it allows any contaminants to get in, primarily microbial contamination, but it could be any other contaminant as well. Another thing to consider is the type of product and the packaging. If it's in an impermeable container, then you're not really concerned with moisture gain or moisture loss. But if it's in a semi-permeable container, again, we talked a little bit about the different humidity condition that we need to consider that when performing stability. So once we have gathered this information, the next step would be to come up with a study protocol. And I've outlined just a couple of slides that discusses what needs to be gathered. It's a good thing to list every different attribute. So you're going to have your product name, the strength, the batch number, the batch size, when it was manufactured, if it's manufactured at a subcontractor, location of that facility, the API that was used, and the lot number for that, who made the API, and the vials that were used, the stoppers, the overseals. And these things differ obviously depending on the type of product. And then the stability storage condition selected, as well as what the final packaging is. Once you have that information, the next step is based on the tables we discussed earlier, is to come up with the specifications. So here you would list each of the different tests that are to be performed, as well as what method would be performed, and then what is the specification for each of these methods. Once you have the methods selected, next thing is to outline in a matrix formation the different tests that will performed at each interval. So as you can see, some tests such as appearance, water content, assay would be performed at each interval from initial all the way, let's say, to 24 months. But maybe some other tests such as particulate matter, seal integrity, or sterility might only be performed at initial, maybe 3 and 6 months, or at some other intervals, normally beginning, middle, end, or some other interval that meets your requirements. Next step is to determine the sample requirements. Obviously, we have the up from initial to 24 months, and each test will need X many samples. Now, in some cases, samples can be shared between tests. So you determine the minimum requirement for each interval and what intervals do not need any of those. You have your total here. And in this case, let's say it's 391 vials needed. At the same time, you want to have extra overage. It's really critical to have extra vials. It could be to perform some investigations if there's a out of specification or out of trend information. But at the same time, if you want to extend this stability at 24 months and you want to now, everything has passed up to that point, and you want to then be able to claim 36 months or 48 months, if you don't have enough extra vials, you're not able to perform the extension. You basically have to start from scratch again. I wanted to talk a little bit about bracketing. This is covered in the ICH. What bracketing allows companies to do is to perform less testing than would be needed. In this example, again, here it goes from 0 up to 36 months, but there are two strengths, strength 1, strength 2. There are 3 batches for each strength. So rather than performing testing at each single time point, you're going to skip some. So in this case, for 3 months, it is only performed at batch 1 and basically batch 2, and then for strength 2, batch 2, and then for the 6 months, there you're going to skip these, and at 9 months, you're going to go backwards and do the same ones that were done at 3 months. So by allowing a bracketing, this allows you to perform testing at a reduced level. In this case, is half reduction of the amount of tests. Some cases with other batches, if you had third strength or fourth strength, you might even do less testing. But you still get roughly the same amount of data, in that you're covering each time point. You're having at least 3 batches, in a sense, tested at each time point. It's a similar concept for matrixing, and in this case, you not only have the container, the strength, but you have different containers. So here you have strength 1, 2, and 3, but you also have a container size A, B, and C. So this would be a rather large stability study if each strength was tested at each container size. And by doing a matrix design, you're able to pick and choose which batch and strength and container size will be tested. And again, this helps to reduce the amount of testing that is needed. I wanted to just quickly cover some of the things that could happen when you have a stability out of trend or out of specification. So things such as hydrolysis, epimerization, oxidation, and so forth. Usually during the initial stability, such as R&D phase, you'll have a pretty good idea that product is susceptible to some of these changes. So you wouldn't normally expect this to happen during the registration batch time, but so it's usually resolved at the early stage of the product's life cycle. A couple of the other things would be some microbial contamination, issues with container and product incompatibility, as well as container closure system failure. So once you've completed and started the stability protocol and you started your stability studies, at some point you're going to have data, and then it's up to obviously the company to make an evaluation. You want to take the data from all the batches, usually minimum of 3, and you want to evaluate any of the test results, and if there's any out of trend or out of specification results, and perform statistical analysis if necessary, and predict if there were any physical, chemical, or biological or microbiological changes. And the data has to be in a nice, clear format. Normally, most stability data is in a tabular format. You could obviously add graphs to it, and usually there's some narrative of any changes and a discussion and a conclusion. Normally, you want to look at accelerated, the long-term, the intermediate, and discuss whether there were any significant or not significant changes of the data. And in many cases, there will be extrapolation of the data. So if you have up to 6 months of data, you would then be doing some data extrapolation to see how long is the shelf life. And it's a little hard to see this chart. It is in the ICH. So this is a decision tree on what to do with the data, depending on when you may have a failure. And this is a chart that shows the extrapolation. So in this case, we have data points from the initial 3, 6, 9, and 12 months, and you're plotting the upper and the lower acceptance criteria and looking to see, at what point is this stop becoming a stable product. So here, I'd say 24 months, maybe a little bit longer, and that'll help you obviously determine your shelf life. For degradation products, you're primarily concerned with the upper acceptance criteria. If it stays below, it's not an issue at all. So in this case, you're looking at again data from up to 12 months, and you're looking to see at what point does this degradation products increase above your acceptance level. Wanted to switch to personal care products as well. And here, a couple of definitions. Generally, products intended to cleanse or beautify are regulated as cosmetics. In the US, they're regulated by the FD&C Act as well as the Fair Packaging and Labeling Act. And depending on the country, some for example, over-the-counter products are considered drugs. In other countries, they're not considered drugs and they're considered personal care products. But in terms of designing studies, it's very similar. The primary difference is that there's no active ingredient in these products. And normally for personal care products, they're performed to a particular protocol or possibly a particular standard operating procedure. In our experience, there are possibly sometimes hundreds of personal care products developed in a given annual cycle, versus there might only be 2 or 3 pharmaceutical products that a company makes in a given year. So it's sometimes easier to have a set standard operating procedure that governs how is a cream tested, how is a eyeliner tested, versus and not have to create a separate protocol each and every single time. Majority of the time, personal care products are stored at 25°C, so therefore accelerated would normally be at 40 degrees, but sometimes these are done at 45, 50 even to speed up the aging process. It is very critical to perform freeze-thaw testing for personal care products, especially products with that contain water. One of the concerns is during shipment of the product that the formulation could separate. You could have crystal formation. So it's a very critical study. Normally, at least 3 cycles of freezing and thawing are performed. And again, in terms of the duration, it will, at a minimum, be 12 months, but it obviously could be 24 or 36 or 48 months as well. And for Europe, it's important to consider the period after opening requirements. That is where the product has to be opened, and how long is it good after it's opened. So it's good to perform that as part of your stability as well, which would be to have containers that are purposely opened, either at once at the beginning or at each interval, and they're tested for the same attributes, in addition to products that are not opened. I'll go over a couple of the tests that are performed on, let's say, a water-based product. So primarily, again, appearance, color, odor, pH, viscosity, specific gravity, solids. If there's some chemical test needed, it could be maybe just the antimicrobial agents, possibly elemental impurities, and then obviously aerobic plate count, yeast and mold, and specific pathogens. But if it's anhydrous products, it's a limited set of tests, primarily appearance and color, possibly water activity if needed, and again, the absence of maybe regulated or banned substances, possibly elemental impurities. If it's a fragrance, again, appearance, color, odor. Sometimes a GC screen is performed just to evaluate the fragrances profile, but most of the time that's not always done. Similar to the pharmaceutical products, you would have the basic information about the product that's going to be tested. So your product name, the batch size, the batch number, the manufacturing date and manufacturer, what the container is, and any of the storage conditions. Again, what's missing from here is the active ingredient, since usually there is not an active ingredient. And again, you would have your test and the methods, as well as the specifications for each. You would put your different tests and the intervals and make a decision on which tests will be performed at each time point and which ones will be performed at less frequency. And then again, you're going to determine the number of samples. Again, for example, for color, appearance, and odor, you only need one. You can do all three with one sample. Maybe for micro, you just need one. Then you can do all the others. So again, you determine the total number of units. And then again, have extra containers in case you had to do repeat testing or you want to extend the interval. Similar to the pharmaceutical data presentation, you would put this data in a tabular or graphical format, list all the results from both accelerated and long-term conditions, and indicate if there's any changes, and perform extrapolation if needed. Usually there isn't anything to extrapolate in a personal care product, but again, if there's any numerical values, you could do the extrapolation. So in a summary, hopefully I've given you information on how to utilize the ICH and the WHO and ISO standards and documents, as well as the information from trade organizations, and then utilize the various R&D information about the drug substance, drug product, or personal care products, and design a stability study to suit your product, and then generate data from those studies and make a determination of shelf life or expiration date. I want to acknowledge Eileen Camacho, Wendy Chang, and Michelle Hubel for assisting me with the presentation. And my contact information is here. I know we'll take some questions at the end. And if you need to get a hold of us, you can either contact Kelly or you can contact myself on any questions you may have. And the next few slides are just the different references that I mentioned. So these are the various ICH references, the FDA, the European, as well as the WHO references and the ISO references. So that is the end of my presentation. Thank you very much for listening.
Thank you, Ranil. Yes, we will go ahead and open up for questions at this time. You can either write out your question in the talent section at the bottom right, or update your status to raise your hand if you want to verbally ask Ranil any questions. We'll give everyone a few moments. And as a reminder, I do want to let everyone know that this webinar presentation has been recorded. All participants of this webinar will receive a follow-up email once the recording is available to view. We post our webinars on our ALS Global website and our YouTube channel. There you'll find a wide range of video resources and our past Webinar Wednesday presentations. Ranil, we do have one question from Angel. It says, do you know if exists a minimum light exposure time period for photostability studies?
Yes, so it's not time in terms of like a month or 2 or 3. It is time. It's in terms of the exposure amount. So I think I don't know the slide number, but it's 200 watts and 1.2 million lux hours. So there's a way to perform that calculation. Generally speaking, that can be done within a few days. So once you put the sample and into the photostability chamber, we do the exposure, one set of exposure for UV light and one set of exposure for visible light, and it keeps track of the amount of requirements for ICH. And usually within a few days, it's finished, and then we can provide that data to the client as well as expose the sample. So that's the normal way of doing it. We do have some clients that want actually real-time, visible light exposure. Normally, this would be kind of done outside, but that's not in the regulation, and therefore, it's some people call it, putting the product on a window. But we had to be careful because most windows these days are tinted, so you don't really get good light exposure. So you'd have to almost do it outside, and we've done that. For a specific client because that's what they require. But normally, most people follow the ICH guideline and do the 1.2 million lux hours and 200 watts of exposure.
Thank you, Ranil. We have a few more questions that came in. What is the procedure for surface water testing for PCPPs?
So if I'm not mistaken on the question, you might be asking about water activity, the amount of water pressure or water vapor available. And so there's an instrument called the water activity meter. It's done by a dew point analysis. And so that, again, there's a chapter in both the USP and the European Pharmacopoeia on performing water activity, and that is to tell if a product is truly anhydrous or not, and that gives you a good indicator whether it's going to support microbial growth or not. So hopefully, I think that's the question. Hopefully that's the answer you're looking for.
We have another question. Is there a requirement on how many extra samples should be included in the stability study?
Not that I'm aware of. So the guidance doesn't tell you to put any extra samples. Obviously, most people do. You can. Here's something to think about. If your minimum testing per interval, let's say you need 10 containers. So if you want to repeat at least one more interval, you need at least, let's say if you have 100 units and 10 is needed per interval, you need at least 10 more. And that's all you can then do. So normally, if there's an out of specification investigation, it's customary to perform some additional testing. It could be duplicative testing, triplicate testing. So if you then need to have additional 10 or additional 20 or additional 30 containers, my recommendation, within reason, is to have at least 30% extra containers. It's unfortunate that at the end, if you don't have extra containers, clients will come to us and say, oh, everything passed. Can I add one more interval? I want to extend this for 36 months, 48 months. We go back in the chamber, count what's left, and there's only 3 3 units left. So there's nothing that can be done at that point. You lost 2 years of time. You'd had to restart your stability to get a longer expiration. So I would say a rough rule of thumb was probably between 20 to 25%.
Thank you, Ranil. I don't see any additional questions.
I see something about sunscreens. Can I answer that one?
Oh, yes. I'm sorry. I did not ask that question. What tests do we need to do for sunscreen?
Sure. For a sunscreen product, obviously it's the over-the-counter in the US, and you have your sunscreen actives. So depending on what the actives are, if you are doing primarily organic sunscreen, so avobenzone, oxybenzone, things like that, you're going to be performing an HPLC assay for those actives as well as any degradants. So that means that the method should be validated prior to starting the stability. If you have inorganic actives such as zinc oxide and titanium dioxide, again, you would be testing for those. They're done differently at the ICP OES or AA. And again, you're going to validate those procedures. So that's the minimum. At the same time, again, you would do probably colored appearance, pH of the product. And again, you might do depending on how the product is packaged, if it's semi-permeable or impermeable, you might do moisture loss or gain. And normally there are water-based products, so you would do microbial testing as well as preservative effectiveness testing. And you would set up that program of up to maybe 2 years and decide the assay gets done at each time point and the other tests get done at maybe less frequency.
Thank you, Ranil. I don't see any additional questions coming in. We did have a few people asking about the recording. I just want to send a reminder that everyone on this recording today will receive a recording of the webinar. We will be sending it out sometime a week following today. So you'll receive it sometime next week. And you can view the recording again. You'll have a link to either our ALS website or our YouTube channel. You can follow us on our LinkedIn page. We'll post future links to our future webinars, as well as other valuable resources and updates. If there's no other questions coming in, I do have one more question, Ranil.
Okay.
It says, it's true that the specification for light exposure is in lux or watts per hour. However, in fact, it is not the same exposure time if I put the sample in the farthest part or in the nearest zones chamber. I'm not quite sure if that was a question.
Well, I think I can try to answer that. And what I would say to that is that goes into how the chamber is validated. So it's important to validate whether it's a photostability chamber or a stability chamber and make sure that the temperature, humidity, and the light exposure is uniform. So normally, you would want to validate that chamber and prove that the light coming from the light source hits the product evenly and not just in one spot. One of the things that you can do is to put the light sensor right next to the product. But if you have sufficient units, you can then put multiple units and you can do not just a single test. Maybe you might do a triplicate analysis on 3 tubes you place within the chamber. But normally, if you validated that chamber, you have pretty good confidence that the light exposure is correct.
Ranil, Angel, I did make your audio on in case you wanted to ask a question directly. Gave me the thumbs up, so I think he's okay.
All right, thank you.
Someone did ask how to decide the number of samples.
Well, it depends on the number of tests you're performing. So I know it's sometimes difficult to have a lot of containers available. Normally, you have separate containers for your microbial testing. You don't normally want to mix up your microbial testing with your chemical or physical testing. So even if you only need 1 unit to do everything, which is not always the case, you at least want to have enough so that the microbiology lab can do their work and they're not relying on waiting for chemistry to finish their work. So you want to come up with that, minimum number, obviously, of units needed. So you might have 1 unit for assay, maybe you have a backup unit. You might have, if you're doing sterility, you might have 10 units or 10 bottles needed for that. If you're doing endotoxin, you might have 1 or 2 units. Usually the method specifies the number of units. So you add up all your methods. What's the minimum? Can you share? So usually the physical test you can share. So you can do colored appearance, even pH, viscosity, possibly all on a single or 2 units. So that'll help you decide the minimum number of units. And remember, you're going to be doing at least 3 batches. So you will have, 200 products from batch 1, 200 products from batch 2, 200 products from batch 3, and that gives you the replicate analysis you need to evaluate how the product does.
Thank you, Ranil. Yes, I do believe that was our last question. Again, if you did want to contact Ranil, in the email that you will receive from the webinar, we'll have his contact information in case you want to contact him directly, as well as he had it on his PowerPoint today. I want to again remind everyone we do send out the Q&A next week. And I want to thank you all for attending today and Ranil for presenting. And I hope everyone enjoys the rest of the week.